this lecture has to do with metabolism and our first metabolic pathway that we will study, and that is glycolysis. This is also known as the Ebden-Meyerhof pathway, and it is also known as glucose catabolism, where we actually break down a molecule of glucose into 2 molecules of pyruvate, the 10-step metabolic process. So first of all, I'm going to dedicate about 4 or 5 slides on metabolism in general. So normally this poster would be about 4 feet by 4 feet, it'd be a huge poster, which sort of encapsulates all the different metabolic pathways that are found in a cell. So shown here is probably easily maybe 3,000, 4,000 different pathways, and all of this must be going on, or at least coordinated inside the cell. So here in all these pathways, you have what are called anabolic pathways. So think of a wrestler using anabolic steroids. They're used to build muscle. So in a similar regard, anabolic pathways are pathways that build carbons. So they build molecules. And the flip side are catabolic pathways, those that break down carbons or those that break down molecules. So glucose is a catabolic pathway because we're breaking down the 6-carbon sugar molecule. A couple of other things here on this entire metabolic pathway or metabolic wall chart is you'll have some pathways that are anabolic biosynthetic, some pathways that are catabolic that break down, as mentioned before. But you also have pathways that cannot go at the same time. As an example, if you want to build glucose, that's gluconeogenesis, that cannot occur at the same time as glycolysis, which is the breakdown of glucose. So an anabolic pathway cannot occur at the same time as a catabolic pathway. That would be a futile cycle. That's just a waste. So an important aspect of metabolism in general is this idea of coordination and regulation. The fact is you don't want a pathway to build or synthesize glucose to be occurring at the same time as the pathway that breaks down glucose. Same thing with cholesterol, same thing with fatty acids, same thing with lipid. So there has to be some fine-tuned regulation and coordination. So the cell must be able to do that. And another important aspect in metabolism is this F word. The F word here is flux, F-L-U-X. Flux basically means is that these metabolic pathways are not necessarily always on. And the enzymes that make these pathways are not always so rapid with such a Vmax, or they're operating at such low Km. It's very much modulated in the sense that when the cell needs it, it's going to pick up. It's going to pick up the pace. When the cell doesn't need it, it will slow down the pace for all these different thousands and thousands of pathways that are found inside a cell. So flux is a very common theme in metabolism. So regulation and modulation, coordination, and then the big word here that encapsulates this is flux. So as an example for glycolysis, you probably want glycolysis in a full-fledged force when you're exercising or you're running away or some sort of fight or flight response. When you're sitting down here, basal, very minimal activity, you don't want your glycolysis to be running full throttle. So flux sort of encapsulates all of these terms. Also, metabolism is kind of like an information superhighway. So there's pathways that sort of crisscross and are going to sort of help aid another pathway. The word that describes this is another very important word in metabolism, and that is shunting. So there's flux, and then there's shunting. The idea of shunting is this. So just as you can, in a huge, interstate highway like this with a lot of bypasses and lane mergers or exits and entrances, you can have a metabolic pathway where some of the metabolites will exit, and it will exit and serve another purpose for another pathway. Or on the flip side, you can have pathway A produce a metabolite and then go into pathway, that particular metabolite can go into pathway B. So it's kind of like lanes. I can imagine if you're a car in this, massive highway in Miami or wherever, Los Angeles. Cars can exit and leave, and cars can enter in and merge. So you want to think of that also in the same regard to metabolism. Another thing I want to mention here is a bad thing. The bad thing is traffic accidents. So what happens if you have, a pileup or an accident? Analogously speaking, what happens if an enzyme is mutated or dysfunctional or defective? Okay, what will happen is that, just like in a major car crash in an interstate highway, you're going to get backup. Well, if there's a defective misfolded enzyme, you also will get backup of a metabolite. The cell panics and it doesn't know what to do. It sort of has this buildup of this metabolite and some sort of toxicity envelopes. Best example of this is something that we kind of study a little bit later in advanced biochemistry or as an introduction in beginning biochemistry. Classic example of this is phenylketonuria, where the first enzyme in the metabolism of phenylalanine is mutated and phenylalanine builds up, and then the cell produces phenylketones or phenylpyruvates, which is toxic. It has cognition effects, and lo and behold, you cannot fully metabolize phenylalanine to its full potential. That full potential also includes neurotransmitters. So the idea of defective enzyme leads to a metabolic buildup. Everything downstream of that pathway gets affected. If you have a major car crash, you have cars piling up, and both are a bad thing. So everything must run fluid. The idea here of fluidity comes in two forms, flux and shunting. You may also want to add a third important theme in metabolism is this control, regulation. Okay? Can't have an anabolic pathway occurring concurrently with a catabolic pathway. That's metabolic waste. Right, so there's literally thousands and thousands and thousands of metabolic pathways in the body, and the first one that normally all beginning biochemistry classes study is glycolysis. So glycolysis is taking glucose molecules and making 2-pyruvate. So that's the important end product here is 2-pyruvate. Another important end product here is 2-NADH, and luckily for us, or for the cell, it also produces 2 molecules of this energy molecule known as ATP. You may have heard of ATP. It's sort of like a currency in the cell in the sense that it could hydrolyze those phosphates and get some free energy. Well, this reaction does not happen in one go. Okay, it's 10 steps. It takes 10 steps to take glucose and split it into this molecule here known as pyruvate with the added benefits of ATP, the added benefits of NADH, that cofactor. So why does the cell why does the cell do this in 10 steps instead of 1 step? Save some time, do this in 1 step. Well, it doesn't do that because of the aforementioned shunting, and another thing is flux. By splitting this down in 10 steps, you can actually produce many intermediaries that can go to other pathways. Furthermore, by splitting this into 10 steps, you produce some high energy molecules. The high energy molecules sort of naturally flow into making ATP. So just from an energetic standpoint, you make a high energy, fully primed molecule, it's going to very easily phosphorylate ADP to ATP. So thermodynamics and then shunting. Another thing is also flux. Okay, so instead of doing this in 1 step where you can, make it go fast or slow, make this in 10 steps. You have better control in making glycolytic flux go very fast if need be, or very slow if need be. Also, regulation is important as well. So you can shut off glycolysis or at least lower the flux at 2 or 3 enzymatic steps in the pathway. So that's the reason, not just in glycolysis, and almost all metabolic pathways that you will study in the future, is this idea of splitting up a reaction into 10 steps or 8 steps or 7 steps or however many steps. Right. Our next introductory slide is why study glycolysis. Well, one of the foremost things in studying glycolysis is that it is in its universal conservation. So you can study the glycolytic pathway in E. coli, and it pretty much is similar, very much similar, even at the 3D level structure of the enzymes. It's very much similar to man. So, the old days when they were trying to funding research, such as the NIH and NSF, they were getting all these funding requests to study bacterial metabolism. Why study bacterial metabolism when obviously you're interested in metabolic diseases in mammals or man or whatever? The idea being is if you can study a pathway in bacteria, more than likely it's akin or it's analogous to what is seen in mammals. So the idea is that we see glycolysis in E. coli, we see glycolysis in bacteria, and we also see glycolysis in us. Very, very important in the red blood cell. Glycolysis is very important in the brain because we usually get most of our energy from hydrolyzing glucose in the brain. We don't hydrolyze fatty acids in the brain, but we hydrolyze glucose, so glycolysis is super important in the brain. And glycolysis is also important in the red blood cell, and glycolysis is also important in the muscle cell. Obviously, you can figure that out when you're doing very, very vigorous exercise. A couple of other points here about glycolysis. There's something here called the Warburg effect. This was probably around the 1900s or so about a century ago. Otto Warburg found out that cancer, actually it produces a high level of glycolytic flux. So cancer is cells that have gone out of control. They have gone into a proliferative nightmare, and they don't die. They've achieved immortality. The problem is when they achieve immortality, they've invaded other cells and they invade the peripheral tissues. It becomes a big problem. Controlling it and not having cell death is a problem. Well, Otto Warburg found nearly 100 years ago that many of these cancer cells actually have a rapid pace, high glycolytic flux of glucose metabolism, that is to say high glycolysis. Furthermore, he realized that pyruvate, the end product of glycolysis, goes to lactate. So many of the cancer cells divert away from the TCA cycle and they produce lactate from pyruvate. So that was something that was sort of a mystery. Why do cancer cells do that? And furthermore, if you can find a way to block this flux from pyruvate to lactate, you may have found a way to sort of regulate or shut down cancer cells. So the Warburg effect is directly related to glycolysis. It's mostly related to glycolytic flux, but it's also related to the fact that after glycolysis, a lot of the pyruvate preferentially and indiscriminately goes to lactate. So you can see here the pyruvate has many different metabolic fates, and that would be a subject of our next lecture, the metabolic fate of pyruvate. So if you're more interested in the Warburg effect and the history of this with regards to glycolysis and cancer biology, please read this review. But you can see here glycolysis and its relationship to all these 4 different categories. All right, a couple of other things here. You're going to hear me mention this word a lot in metabolism, and that is the word called flux. So flux, shunting, regulation, regulatory enzyme, all of these are synonymous with the whole concept of metabolism.